Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Started with the next session. It's about complex biological systems, and our speaker today is Langley from Arizona State University.
So first I want to, is the sound okay? It sounds weird to me. It's all right? All right. So first I want to just mention that this talk is not about data-intensive science. It's about model-intensive science. I think that that they're both important, and that we can't forget about the models in the excitement about the wealth of data that's becoming available. And in particular, I've been working in domains where data is not very common. And the one I'll talk to you about today, there is data, but it's very diverse and very hard to get. And I think that many of the challenges are in the complexity of the models, not in the size of the data sets. Lots of other people who have contributed to this over a couple of years. Let me make some general comments first, and these are about challenges in scientific modeling. This is about system science and system-level models. And one of the things that really distinguishes science from other endeavors is that there's a long history of using communicable formalisms. That is, formalisms, ways of describing models that people can communicate over long distances. Previously in journals, now I guess through the web. But science is inherently a community-based process, and exchanging the content of models is important. And we can't ignore that. I think much of the work on data-intensive science does ignore it and builds very shallow, opaque models. Second, science is concerned with not just summarizing data or making predictions, but really with explaining observations in terms of familiar concepts and familiar mechanisms. So it's, again, moving beyond the data to try and come up with some kind of a causal or other explanatory account. Third, science doesn't operate like a batch data mining algorithm. You don't give it the data and then out comes the answer. It's a cumulative process. It happens over time, where you extend the models gradually as you fill in technical details. These are things that all of these tend to be overlooked in the rush to do data-intensive science, and I think that we should not forget that these have been part of the scientific process for centuries. They're going to continue to be because science just works this way. Now, if you're going to build software environments for scientific modeling, you have to address all these issues, and in this talk I'm going to tell you about one such environment. It's about aging, and I think that aging really is the ultimate problem in systems biology. It's a very complex set of interacting processes. We know that there's at least 7 or 8 basically different causes underlying aging. Each of them is complex in its own right. I'm talking about things like the mutation of mitochondrial DNA, the accumulation of lipofuscin in lysosomes, the cross-linking of proteins in the extracellular matrix, and shortening of telomeres in the nucleus. But there's more. And again, each of these is a complex set of interacting mechanisms, fairly localized in different parts of the cell. And then there's, of course, tissue level and whole body level aspects of aging too. It almost demands computational assistance. Okay, we can try and do it all by hand, but I don't think you want to. Here's an example of what happens when you try and do it by hand, with the exception of you're using a drawing package. John Ferber is a biology friend who has been trying to develop system-level models of aging for the past 10 years. He reads the literature all the time. He's been building this network model, and this is just the top half. It goes this is the cellular level part of his framework. The bottom level is tissue level and whole body stuff. And let me just expand the top left corner. That's the compartment for the lysosome. You can see that there are various things going on. There's lysosomes disassembling junk protein that's producing reactive oxygen species and also free iron. Together, those lead to cross-linking that forms lipofuscin. That's a kind of junk that starts to fill up your lysosomes. That makes the lysosomes less effective and can lead in turn to more reactive oxygen species, and so on. It's a fairly complicated story. This is really, even though this is an explanatory model here, it's I would say still not very detailed, still fairly superficial. It's also informal. And so these are just a bunch of boxes with text in them and links with some of them are inhibitory, some of them are excitatory links. They get at some of the intuitions that biologists have about the effect of lysosomes, the role of lysosomes in aging, but they don't really have the kind of character that you want in order to really be precise. If you want to put this on the computer, you want something that you can really interpret, that the computer can interpret, and that we can interpret. Now, of course, the systems biology movement and computational biology more generally has many candidate formalisms. There's a whole bunch of them. They tend to come out of different traditions. Ones that come out of physics tend to be differential equations. There's Boolean networks. There's Bayesian networks. There's a whole bunch of others. There's the pi calculus. I taught a course at Stanford on this some years ago, and there were at least 10 different modeling paradigms that you find in the literature. I think that they each have strengths and weaknesses. Differential equations, unfortunately, require both functional forms and parameters. Boolean networks require assume discrete variables rather than the kind of continuous ones that are usually measured with respect to aging. Bayes nets have some nice aspects. The graphical structure tends to reflect a biologist's intuition, but they have these probabilistic parameters which you either have to make up by hand or you have to have enough data to estimate, in this case, we really don't seem to. So my view is that we need a notation that makes close contact with biologists' ideas about aging mechanisms and that is somewhat simpler than these. Now, somewhere along the way, probably back in the 60s, people got the idea that computers were good for numeric manipulation, for number crunching. But they aren't. They can do that, but they can also do general symbol manipulation. So pretty much anything you could take any scientific model you want, in any formalism you want, and you can find a way to encode it on a digital computer. And so in biology, for example, you can say, well, if we want to represent the structures that occur in organisms, like the structure of organic molecules, we can do that on the computer. If you want to have processes like biochemical reactions and pathways, we can encode that too. There's a project at SRI led by Peter Karp that's been going on for many years where he's got, it's called MetaPsych, and he's got a whole bunch of metabolic pathways stored on the computer. And so we can do that. The problem with actually building metabolic networks for aging and, say, gene regulatory networks to influence aging is we just don't know enough. I think it would be very difficult to actually draw all those out. So I think what we need, for now at least, is a more abstract representation, similar to the kind you find in the artificial intelligence literature on qualitative reasoning. And I'm thinking of work by Ken Forbes and Ben Kuipers. It's been around for a few decades now. This is the particular approach that we've been taking. It's very qualitative, it's very abstract, but I think it's the right level for what we have to work with now in terms of content in the aging research community. We've got places in a cell, things like the lysosome and the cytoplasm. We've got quantities that can be measured or have been measured in those places. These are for both unstable entities like reactive oxygen and free iron, also for stable entities that accumulate over time, things like oxidized protein and lipofuscin. Okay, and my take from talking to biologists is this is a fairly reasonable, useful distinction for a certain level of description. So those are the quantities that occur in certain places. You can have the same quantity being measured in different locations, like in the lysosome or the cytoplasm. And you've got causal influences or relations between quantities. These can say things like, well, if you increase this quantity, then this one's going to go down, or this one's going to go up. Now, those quantities, they could be things that are directly measured. They could be sort of rates of change, first derivatives. Now, one of the things that's nice about this level of abstraction is that you can also state these things in a constrained form of English that I believe biologists will find useful. So this is actually a programming language that we have that you can type these things in, or you can put them in a file and load them. Here I've loaded in a file, a model of what's going on in the lysosome, and it's got three places: the cell, the lysosome, which is in the cell, and also the cytoplasm. It's got 11 quantities, including one degradation rate, but also 2 instances of junk protein in different locations, reactive oxygen species, 2 lipofuscins, and so on. And most interesting is these hypothesized claims. These are the causal relationships. And again, these are stated in English too, but you can see that they're really just, you can think of them, they're connecting 2 of these quantities with a qualitative relationship. Okay? It says that junk protein decreases with the degradation rate. ROS increases with iron in the lysosome. Lipofuscin increases with the damaged membrane. Okay? And there's the stuff in red in brackets. Those are just text notes to expand on that. That's not actually part of the modeling language. Now you can draw this out. This is not part of a graphical interface. This is just a depiction of how you might display these. We are working on building a graphical interface for this, but we haven't done it yet. So this is the equivalent of the text that I showed you. And so you can see that these are supposed to be quantities that one could measure. They don't all have to be measurable, but they could be measurable. And there are causal connections between them. And you can imagine taking steps along here and using this to make predictions. Okay. Here's a mitochondrial model. These are just the claims, not the places or quantities. Okay, so it's a little bit simpler than the lysosome model. This is about how mutations occur in mitochondrial DNA and what the effect is. This is a model of what's going on in aging in the cell nucleus and the shortening of telomeres. And again, all of this is taken. It's a sort of an attempt to formalize what was in Ferber's informal model that I showed you earlier. These are all, though, have a clean semantics, and you can store them in the computer, and you can do operations over them. Now, so that was the first half. You have to be able to represent this content, and you have to do it at a level of abstraction that makes sense given the available results. One thing I meant to mention but didn't say is that the literature on aging has many different kinds of data. Some of it is from controlled laboratory studies, right, where somebody does a knockout study with C. elegans. Some of it is where they do experiments where they do caloric restriction on rats. Some of it is genome-wide association studies in humans. Some of it is pathological studies of deceased organisms. So it's all over the map. And one of the reasons why I feel strongly that a qualitative language is the right thing now is that that's the level at which you can bring these together. So in some sense, describing things at a qualitative level is a response to the heterogeneity of the data and is something we're thinking about maybe in other fields where the same thing happens. Now, that's representation. Now let's talk about reasoning. Okay? I would claim that a scientific model of aging or anything is missing something big unless you can relate it to observations, to the phenomena. And you want it to be able to answer questions like, well, what biological effects does the model predict? What observations or experimental results does the model match? How does the model explain a given phenomenon? Not just what's the prediction, but what is the chain of reasoning that led to that? And how, if you change the model in various ways, what would that do to your predictions? Sort of to do thought experiments. So the intuition here is that you can do this manually. People have done it manually. But when you start to get to the kind of complex models you need to have a real theory of aging, you really need computational support. Now, if you're going to do that, you have to have some way of representing the predictions. And what we do for that is we use a notation that's very similar. Well, you have to represent the predictions, you have to represent the observations, and we use for that a notation that's very similar to the model hypotheses. Basically, we say you can say some quantity increases or decreases with some other quantity. For example, lipofuscin increases over time, or lytic enzyme decreases with reactive oxygen species in a particular location. Or you can say, and this is different, that a quantity does not change. So there's no experimental effect. Now, this is assuming that all other things are held constant, right? That whether it's experimentally or statistically, the assumption is that somehow you've got a result that says that if under these conditions, if you increase this, then this is what happens to this other variable. Again, you can say these in constrained English. And the important point here, and I've actually had some trouble with a few biologists getting this idea across, is that these are the phenomena that the model is supposed to explain. Okay? It's not part of the model itself. So you can actually have the same statement as an observation, and that's not part of the model. It might actually, the same statement might appear in the model, in which case the explanation is trivial. But it's not the model, and it's important to keep them distinct. So I talk with Ferber about this, and I say, we got to have these separate. He goes, why? You just throw it all together, because to him, it's all part of the story, which in some sense it is. But I think that when you get to predictions, the observations that are not directly predictable, where you have to do reasoning over it, then it becomes more interesting. So, okay, so you can do the same kind of thing with these. You can store empirical facts that come from the literature about changes in time, about co-occurrences in variables. You can ask questions like, well, does lytic enzyme change with ROS? Or oxidized protein change with R222? And the system will come back with an answer for this. In one case, it says it decreases. In another case, it says I predict it should not change. You can also ask it to make predictions with respect to the empirical facts that are known. What the system does not let you do is go off and make all possible predictions, because there are just going to be a shitload of those. You can cut that out. And that really the only predictions you should bother to make are the ones the user asks you to do, or the ones that are directly relevant to observations you have. Otherwise, your user is going to be overwhelmed. It's not that you couldn't do it. It's just that I don't think it's going to be useful. There are some simplifying. So let me tell you. I'll tell you in a minute how the system makes these predictions. It's not going to surprise you too much, but I want to be clear first on some simplifying assumptions that we are making in order to do this. Okay, so the formalists out there would want to hear this, because the reasoning methods we have only make sense with these assumptions. One of them is that all the quantities, remember, so we don't actually represent the numeric values. Okay, we just we have the variables that stand for the variables. It's like in algebra, but we're going to assume that all the quantities are positive, including ones that describe rates. Yeah, Frank. Okay, non-negative. Sorry, and you're right, and so you don't lose any generality with that. Also, so the system that we're modeling exists in a single qualitative state. What I mean by that is that a quantity is going up, or it's going down, or it's staying stable, but it's not doing anything else. Now, that might seem like a crazy assumption. I think that it is in some domains. If you were trying to model, say, oscillation in a population ecology, that would not work. But in something like aging or in Alzheimer's disease, things just keep going pretty much in the same direction. And I think it's a pretty reasonable modeling assumption. It does greatly simplify things. I do have ideas on moving beyond that, but in the current system, that's what we assume. Also, if nothing influences a quantity, then it has a constant value over time. Stable quantities, remember that distinction? They accumulate effects on them over time. So if something is increasing them, they just keep increasing. Transient quantities do not accumulate effects. And so this is key to how the system reasons about temporal phenomena. Again, these I think are relevant and reasonable for aging, age-related diseases, but certainly not for all biology and certainly not for some other fields. Now, how does the system make predictions? Well, you give it a qualitative query about two quantities. For example, does X vary with Y? And it takes the thing the dependent variable you're trying to see is affected, and it chains backward from it. And it finds all the paths through the network that connect the dependent variable with the independent variable. Now, if there's no paths, it predicts no relationship. If it finds a path and there's an even number of decreases links, that's like minuses on the links, then it says there's a positive relationship, that is D goes up with I. If it's an odd number, it says it decreases. Okay, it's just multiplying the signs. Yes.
Do you have explaining away?
Remind me what that is.
Two causes.
Just a second. Just a second. Now, if there's multiple paths and all the predictions agree, no problem. If they disagree, well, so the current system says, well, it's ambiguous. But there's more on that in just a second. So this is really a kind of qualitative reasoning about how variables change in response to experimental manipulation. Here's some abstract examples, right? So the top one, you've got an even number of minuses, so you say that D increases with A. The second, you've got an odd number of minuses, so D decreases with A. The third, D does not vary with A. Here, the fourth, there are two pathways, but they agree in their predictions, so it says they increase. And this one at the bottom, it's making different predictions, the two pathways, and so it's saying they're ambiguous. Now, the way that we handle ambiguity is it comes out of something that Jeff Schrager and I did with respect to gene regulation back about 6 years ago. We say, gee, and this is what we think biologists do. We say, well, it must be that one pathway dominates the other. And that becomes now part of the model. Now, is that explaining away? Right. That's right. So this is explaining away as an idea that is associated with the Bayes net literature, right?
It's associated with Bayes net, but it's a fact of life that you have two causes for the same effect. Once you get evidence for one cause, the probability of the other cause goes down.
Right. Okay.
So there's a backward influence, backwards to the causal chain.
Right. Well, we should talk about whether there's a way to fold that in here while remaining at the abstract level that I'm trying to. Yeah. But anyway, the intuition here is that this now becomes part of the model. Some qualitative modelers don't like this idea because they want to have everything be local, and this is sort of a global characteristic of the model. But that's what we do, and I think it's a reasonable thing to include. We can do the same kind of thing with temporal predictions. I'll skip over this, except to point out that it's really the way things work differently has to do with whether or not the beginning of the chain, if there's an unstable quantity in the chain, you can basically ignore everything before that and just start with the first stable quantity. And you assume that that's a constant value and that things follow from that. Remember, the stable quantities accumulate over time. All right, explanations. So if the system has made a prediction, the user can come back and say, well, explain that prediction to me. In this case, it's called P1. It's lipofuscin increases over time. And here the system has printed out 3 different explanations, and those are just the pathways it found, right? So no big deal about this. But let me just show you what those look like graphically. This is one of them. I think that's the second one, right? So lipofuscin increases with junk protein. And here's another that's more interesting. This says lipofuscin increases with ROS. This has a loop in it. And so whenever the system hits a cycle, it just stops and says that that's the starting point. And I think that's the right thing to do. There's also interesting things. You'll notice that there can be positive and negative feedback loops. Positive feedback loops are not an issue. Negative feedback loops are things that I believe you have to do something like state that one of the subpathways dominates the other, and then you can tell whether it's really a negative feedback loop or not. That's not implemented yet, but we're working on it. Okay, so I told you there were 3 challenges in building complex scientific models. One was representing. We talked about that. Another is reasoning or interpreting the models, and I've just finished talking about that. And the third is updating those models in a cumulative way. You want to be able to extend, revise the models easily and efficiently, and the framework that we've got lets people do, well, at least in principle, lets people do that in a very nice way because it's a very modular representation. You can throw in new quantities, places, links, and you can add you can add notes to things. You can remove them. You can disable or enable an empirical claim or a hypothesized claim or an empirical fact. You can save revisions. So it's this modular character that makes it pretty straightforward to do using the same kind of syntax that I showed you for describing the models. So for example, if you wanted to look at what happens if you revise the model, here's some predictions and some facts and associated predictions, and you'll see that the first 2 has ambiguous predictions, but if we then go in and disable one of the hypothesized claims, C1, then if you run it again, it makes different predictions. Now the model is consistent with those 4 phenomena. So you can do those kinds of sort of mental experiments on changing the model to help you explore the space. In principle, of course, we could try using machine learning techniques to search the space of models. I'm certainly think that's worth doing at some point, but I thought it was better to first get the basic system down and to build an interactive version before we built that automated model constructor. Okay, status of the system, still under development, but it already lets people load, examine, and alter qualitative models of aging, answer questions about how one quantity affects another, compare the model's predictions to facts, as I've shown you already, and to determine the effects of changing the model on those predictions. And we've got initial encodings for 4 of those portions of Ferber's diagram, for the lysosome, the nucleus, the mitochondria, and the extracellular matrix. We have not implemented pathway dominance, but I think we've got the basic machinery to get reasonably complex models of aging. Not all the biochemical details, right? I mean, I've heard some people say things like, you can't have systems biology unless you've got quantitative models, which I think is nonsense. I'm not against quantitative models if you have enough data to support them. I've given talks in this workshop on quantitative models in ecology. I just think that in this particular field, we're not ready for them. Generality. So I think this will work not just for systems biology of aging, but for any domain where you have very diverse results that basically require abstract qualitative models that still move beyond the data to provide explanations. And where scientists have at least some informal hypotheses about the causes of the phenomena, and this last clincher where the temporal effects are primarily monotonic, where things are going in the same direction over time. For example, many age-related diseases like Alzheimer's, Parkinson's, and many types of cancer. Lots of intellectual influences here, both in computational biology and qualitative AI, reasoning and AI, languages for scientific simulation, and certainly probably the biggest influence has been Jeff Schrager's work on BioBike, which is a biological analysis environment. But it puts together ideas in different ways. Okay, pretty much last slide. So we're still in the early stages. We still need to, for example, build a graphical interface for viewing and using the models. I think it would be useful to group the causal influences into processes that stand or fall together, as we did in some of our work on ecological modeling. I do have ideas on how to make these links conditional on the context in which they occur. For example, what tissue, what organism, and so on. And also stick some arithmetic conditions on them to try and overcome the monotonicity assumption. We certainly need to evaluate its usability and apply it to other age-related diseases. Probably the thing I'm most excited about, though, and that we're most actively working on, is making this available remotely over the web to support not just development of a model by a single user, but by a community of users. Something like Wikipedia, but that moves beyond the text notion in Wikipedia to formal models, scientific models that are built by the whole community. I think that has great potential and is a very different model that could speed up model creation. Okay, to summarize then, we've got an interactive environment for biological modeling. It supports abstract but communicable models of aging that specify relationships among biological quantities. It can interpret those models in ways that answer queries, that generate predictions, and that go beyond that to actually explain the phenomena in terms of causal chains. And it supports the cumulative extension of those models so that over time you gradually improve their coverage, and I think it will be able to do that with little training and effort. So that's it. Questions? Thank you.
So I don't have to repeat your question.
Yeah, so I understand that your model is qualitative.
Yes, did I say that?
But I know that you put in the caveat, but I think that in order for it to be useful as
Why do you believe that?
I'll give you an example.
Uh-huh.
Certain molecules at the lower quality actually would inhibit or actually promote the expression of genes, and then the large quality.
So yes. So I think there may be examples of that.
That's actually very important. It's a very
Well, I know it happens in some parts of biology. Whether it's an effect in aging or not, it's an empirical question. I don't know of any results like that, but there may well be some. So yes, so I would say that there's a compromise position that addresses that question, but doesn't go the full way, and that's to build a semi-quantitative model. As I was saying at the end, I have plans for introducing arithmetic conditions on these links. They would say things like, if this variable is greater than this threshold value, or if this particular arithmetic combination is greater than this threshold, then the link is active. Otherwise, another link is active. And that would get at this kind of different conditions without having something like a differential equation model. I think it's more tractable, and you could get by with relatively small amounts of data to support that.
Right. The second question is, actually, you're describing mostly causal relationship for biologists, and I think studying them and confirming them is actually it's quite tricky for them
Sure.
as well. And each new causal relationship discovered is maybe a Science Nature Cell paper.
Sure.
Most often, you also see those non-causal associative relationship.
Mm-hmm.
So I was just wondering, what's the implication of putting
Well,
them in?
so the modeling environment doesn't take a position on that, but I think if something is a very strong experimental result, then I think you could certainly put confidence levels on different parts of the model, right? So you could have a hypothesized claim, and if it's really tied in directly to literature, you could put a high probability on it. If it's something that is more hypothesized, you stick it in as connective tissue to explain something you've observed and to complete a causal chain, then you might have a lower probability on it. But I do think that this happens in science. People come up with ideas for things and they don't have direct evidence for it. They may have, and it may be something you can't even measure, but that doesn't mean you don't think that it's really going on. So I think that that's a legitimate point, but I think that we can have both. David. Yeah.
Have you done the knowledge assessment yet on the dominance of one pathway over another?
What do you mean by knowledge assessment?
Either reading them off from a paper, or have some expert tell you which one dominates.
No. So really, what we've done so far is worked from Ferber's network diagram, and
whether
you call him an expert or not, I don't know. I mean, he's been reading the literature, avidly for 10 years and really knows a lot of the stuff. But no, we have not done that analysis.
Okay. Do you think he could pull it off?
You mean to say whether or not?
To make good judgment calls about which. I'm just curious about how much dominance there is.
I don't know. I know that we've got some loops in them in his networks, and in the qualitative. Again, the nice thing about quantitative models, if you can get away with them, if you have enough data to justify them, is that issue goes away, right? In the stuff we did on ecological modeling, we had loops all the time. But if you know the parameters and the functional forms, then who cares?
Right.
But when you're trying to work at this qualitative level, then it is an issue.
Right.
No, I don't. But I agree with you. That deserves more attention.
Thank you.
So it looked like a lot of the predictions and things that you were making were fairly local, either at one scale or another. And
Yes.
the model that I just perused briefly on the web was multi-scale. How well do you think this is going to do?
I think that
Crossing scales.
that the abstract qualitative character of these models will make it very easy to make multi-scale predictions. Again, I think they'll be fairly weak predictions, right? So, for example, even if we extended this to have these arithmetic conditions on things, we still wouldn't be able to predict the actual response, right? It would be able to say, suppose we were trying to do some experiment with a model organism, and we wanted to do some, look at the effect of caloric restriction or or rapamycin or something like that. Okay, this will tell us what direction things will go, but it will not tell us how much. And that's I think that's okay for now. But yeah, I think that it's just a set of links. And so if the links happen to cross level boundaries, I don't think that that's an issue as long as there's a direct link. So if you're now talking, if you're talking instead about having a model at this level of abstraction, a separate model, and then somehow going from one to the other, that's a whole different story. But as long as you're willing to have explicit connections, which Ferber does when he goes between the levels, then there shouldn't be an issue. But it may not be satisfying to people who do multi-scale modeling. I think that when they talk about that, they're usually talking about somehow different levels and somehow automatically going from one to the other, which is not this would not support, that I can see. Anyone else? Yes?
I just wondered whether you'd looked at any of these sort of fashionable knowledge representation formalisms like OWL, these semantic web formalisms.
Oh yeah, that is fashionable.
And if so, could you comment on why they might be suitable or why they're not suitable for this sort of thing?
I'm sure that we could redo this in OWL. I think that some of the critiques of OWL are it doesn't have full representational power, but for some things. But these are remarkably impoverished. Although this is a causal explanatory formalism, it's not very rich as far as sort of first order logic goes, right? So I'm pretty sure we could redo it in OWL if we chose to. But this is all written in Lisp, and because it's a good language for rapid prototyping. If we wanted to redo it to make it more, I don't know if putting it in OWL would actually make it more accessible to people or not, right? I mean, my goal would be in building a website that would support all this, is to make all that transparent to people. People just go and not worry about what's going on behind the scenes, and give them the ability to create models and not even see what the internal representation is. To either have the graphical or the natural language front end to it, because at least how many biologists are going to master OWL? Right. So, yep.
So let's thank our speaker again.
Right. Thank you. I didn't mean.